Welcome. My name is Ariel Louwrier, and I work for a company called StressMarq Biosciences based in Victoria, Canada. Today I'll be giving a talk about the effects of sonication on a-Synuclein preformed fibrils. I would like to thank the meeting organizers for this opportunity. We will start by looking at a brief introduction of a-Synuclein fibrils, followed by current best practices of using them. We'll then examine some of the key aggregation parameters, and then look at some of the work we've done with sonication and solubility. We'll then conclude and take any questions from the audience. A-Synuclein is a small 15 kilodalton protein whose physiological function is not well understood. It can act in a prion-like manner and can generate large families of oligomers and fibrils, sometimes referred to as aggregates. Fibrils are toxic to cells and can be processed by them, including C-terminal truncation. A-Synuclein can also go on to generate Lewy bodies and neurites, which are structures that are associated with Parkinson's disease and Lewy body dementia, collectively referred to as synucleinopathies. This disease state has no known cure. How does the aggregation happen? Although not fully elucidated, it is known that unfolded monomers can associate with membranes, and then depending on the environment, as well as any mutation, such as the A53T familial Parkinson's mutation, and any levels of truncation and macromolecular crowding will allow these monomers to associate with each other and begin to stack themselves into oligomeric structures. These can then go on to form longer protofibrils before becoming long, mature fibrils having associated with each other. These mature fibrils, however, can also go on to break and self-seed this very same event, leading to an acceleration of fibril construction. In terms of their toxicity impact, this happens in a variety of ways on neuronal cells. A-Synuclein is known, for instance, to be associated with presynaptic vessels. It's also known that in the presence of dopamine and its oxidative downstream reaction products, that these can form toxic oligomers, which can go on to poison the cell. They can also adapt a pore-like structure, which disrupts vesicles and membrane integrity. This can be particularly relevant in terms of mitochondria, where it can go on to create mitochondrial membrane depolarizations. From a trafficking perspective, a-Synuclein exposure is also known to upregulate GRP78. This cartoon gives us some insight into a potential mechanism how these structures occur. Initially, we start with an alpha helical structure, which then goes on to change and go through a variety of different states until it creates this beta sheet structure at the end. This is particularly useful from a reaction kinetic perspective because the addition of thioflavin T allows us to monitor this reaction particularly easily, designated here by the blue line. If, for instance, you are interested in disrupting this aggregation behavior through the use of an inhibitor, then you would be able to visualize this lack of aggregation rather easily by suppressing this curve. What type of species are involved with a-Synuclein fibrils and oligomers? There are a variety of different preparations of monomers, which we refer to as type 1 and type 2, which unsurprisingly go on to lead to fibrils of type 1 and type 2. Oligomers themselves can be various. They can be roughly spherical in size, approximately 25 nanometers in diameter, or resemble small twisted pigtails. Filaments look rather similar to fibrils. However, they are soluble, likely protofibrils, and look like thin fibrils under electron microscopy. In terms of fibrils themselves, the building blocks thereof can be type 1 or type 2 or even a type 3, which we'll examine shortly, and come from either mouse or human species, and can also incorporate mutations such as the A53T mutation. What's important to note is that they have differential properties. From a visual perspective, looking at human alpha-synuclein preformed fibrils and mouse, they look very, very similar. There's nothing really to show any differences, although mouse alpha-synuclein preformed fibrils tend to be more aggressive in vivo than human ones. Again, from an EM perspective, if we look at type 1 and type 2, which are the two main types of fibrils that have been investigated in the literature, and compare it to the familial mutant A53T in the bottom left-hand corner against the filaments, which are the soluble protofibrils, which do not seed, visually, they look very, very similar. However, that said, what happens if we try to carry out a seeding reaction with these fibril or filament constructs? You can see on the purple line that if we use a type 1 fibril, we get a very steep increase in thioflavin T, which means that the beta sheet structures are highly prevalent. However, if we do exactly the same thing using a type 2 fibril, we get a much, much more depressed signal, approximately 20 to 25% of the type 1. So clearly the beta sheet structure is quite different to the type 1. What happens if you seed primary cells with both of these structures? On the left-hand side, you can see the evolution of phosphoserine 129 pathology. And it's very clear and obvious on the left-hand side in this middle slide in green labeled with FITC here. On the right-hand side, using a type 2 fibril, however, you get almost no pathology whatsoever. In fact, it's just background. So again, despite looking very, very similar, the outcome of using them is very different. From a type 1 perspective, which really does describe the majority of preformed fibrils that have been used and examined in the literature, presumably because of their prevalence of generating serine 129 pathology, phosphoserine 129 pathology, you can see that having been injected here into Sprague-Dawley rat brains, you can see that after just 30 days, there is an evolution of phosphoserine 129 pathology, and not just at the injection site, it travels across the brain, so transport occurs as well. Given how long these diseases occur, it is well worth understanding, of course, that in this case, in this model, the pathology is generated in just 30 days. Let's have a look at the current best practices. These are largely described in the literature associated with the citation given at the bottom, provided by the Michael J. Fox Foundation. There is a requirement to sonicate the PFFs directly before use. The active component itself is unclear whether they're shorter fibrils or oligomers. However, it is given that the fibril size should be 50 nanometers or smaller to generate the phosphoserine 129 pathology. There is no negative impact associated with an extended sonication time and no differences in the amyloid conformation or fibrillar structure. Shorter fibrils seem to be more efficient at seeding, and sonication heat may negatively affect fibril stability, so cooling is required during the process. So the known effects of sonication are a reduction in the size of the fibrils, declumping of any fibrils, and a significantly higher activity when these fibrils are then added or used in vivo later on. Our question is something else occurring? Fibrils themselves are insoluble constructs, and the monomers, on the other hand, are soluble. So we are using components in two different phases. So here's a proposal. What happens if we can remove the two-phase system and instead generate a single-phase system? First of all, it is likely that any unfavorable energetics associated with substrate desolvation would be eliminated. If those limitations were removed, by creating the single phase system, we would expect to see an increase in the prion-like catalysis, because we would expect to see an increase in the ability of these molecules to associate with each other. And of course, this is known to occur. So does sonication create a single phase system? Well, we set out to have a look at this. Here are some micrographs that are representative of a variety of different levels of sonication on preformed fibrils. On the left-hand side, you can see a control where the mean size is just under 70 nanometers in length of these preformed fibrils. As sonication is increased, given at the bottom here, where the amplitude is increased from 20 to 40%, we see a decrease in size of these PFFs. Now that is known to happen and has been shown in the literature before, and here we have a graph that shows roughly a linear decrease in size of the PFFs as the level of sonication is increased. However, what happens if we look at the level of solubility? On the left-hand side, prior to any sonication, you can see in the two leftmost lanes, the supernatant and pellet here, that the overwhelming amount of fibril is associated with being insoluble. However, at the maximum level, shown here on the right-hand side and in red, the maximum level of sonication, this has moved completely to the soluble phase. So what happens over time? Is this a transient effect? In other words, if you carry out sonication and leave your sonicated materials, for instance, in the freezer for days, weeks, or months, is that solubility maintained? And interestingly, it is. You can see at this high level of sonication that even going from July to October, over a period of months, being stored at -80 degrees, that the amount of material that is in the soluble phase is the overwhelming vast majority. So clearly, the effects of sonication in terms of solubility are maintained for long periods of time. What happens in terms of a seeding THT assay? If we take a fibril and carry out a normal seeding assay, such as the ones you've seen before, this is represented by the purple line in the middle, and we then sonicate at different levels. Here we have 3 sonication cycles followed by 6 sonication cycles. Very similar outcome. We can see an approximate doubling of the seeding outcome. The bottom lines here are associated with a variety of different controls, but clearly the impact of sonication allows the seeding activity to increase. Now, is that a function of the fibrils themselves? And the answer here is no. So if we just take the fibrils themselves and see what their THT binding looks like, as we increase the level of sonication, we actually decrease the amount of THT binding. So clearly it's what they do and not them themselves that is important in this particular case. This, of course, would have a large potential impact also when trying to measure the level of fibrils that might be present if you were using THT to do that. In conclusion then, preformed fibrils are reduced in size during sonication, but the size reduction alone is unlikely to be responsible for the solubility differences. The fibril size, however, may still be a good indicator of increased solubility after sonication. The sonication of the preformed fibrils results in those fibrils moving from the insoluble to the soluble phase, and it is likely that the dramatic increase in solubility removes or reduces the limitations of the prion-like activity. And these limitations may be due to the unfavorable energetics of substrate desolvation. The best way to identify if fibrils are properly sonicated is to do a sedimentation assay and see what is in the soluble or insoluble phase. Once sonicated, fibrils may stay in solution for at least 3 months at -80 degrees. Sonication-induced oxidative free radical modification of the preformed fibrils does not occur. This is from data that we haven't shown simply due to the length of this talk and is not responsible for increased solubility. Sonication can decrease thioflavin T binding of fibril seeds. And sonication can significantly increase in vitro seeding activity of preformed fibrils in seeding assays. I'd like to acknowledge the following 3 individuals who did the majority of this work in our laboratories: Alexandra Netter-Glangau, Sophie Sun, and Vicky Meyer. For any other information, please feel free to get directly in touch with us, or you can email questions to our sales coordinator given here, and also visit our landing page given at the bottom. Thank you very much. I look forward to any questions you may have.